share my screen?
Yes, of course. Lakshmi, are you
Oh,
there?
yes, yes, I'm just
Can I help you somehow?
uh, yeah, my screen is visible to all of you.
Yes, 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 of course. So you can start. Thank you.
Screen is changing.
Yes, of course.
Okay, so here we go. So good afternoon, everyone. I am Dr. Lakshmi Shaw, currently a postdoctoral scholar with Texas State University, USA. Pleased to present my research on the topographical assessment of neurocortical connectivity during meditation, focusing on two frequency domain measures, the directed transfer function and partial directed coherence. So this work was conducted at Indian Institute of Technology, Kharagpur, during my PhD, and this work has already been published actually in Springer Cognitive Processing. Yes. So here is an overview of today's presentation. I'll start with the motivation and background, then describe our methods, including data acquisitions and analysis. We will review our results, discussion, and their implication, and a few limitations too, and finally finish with concluding remarks. So the recording of the electrical potential produced by the synchronized firing of neurons are mostly captured as an electroencephalogram, that is well known as EEG. So among all the brain imaging techniques, EEG is unique due to its low cost, simplicity, accessibility, high temporal resolution, portability, and non-invasive nature. So meditation, particularly here I'm talking about Indic meditation, that is Kriya Yoga, is known to be induce distinct changes in brain dynamics. Our objective is to assess how meditation affect directed neural connectivity using directed transfer function and partial directed coherence as our primary measures. So understanding brain connectivity is crucial for mapping how different brain regions interact. Functional connectivity refers to statistical dependencies, while effective connectivity addresses causal influences. So, however, EEG analysis is complicated by nonlinearity and volume conduction, which can obscure true connectivity. So, this underscores the need for robust and quantitative measures. So then comes to Kriya Yoga meditations. So whenever I'm saying Kriya Yoga, which is an ancient meditation practice involving breath, mantra, and mudra aimed at deep concentration and well being. So, EEG captures brain activity across various frequency bands, each associated with different cognitive states, and meditation produce unique signature in these bands, reflecting altered connectivity. So our applies directed transfer functions and partial directed coherence to EEG data collected during Kriya Yoga meditations. We quantitatively compare these measures using absolute energy, signal to noise ratio, and relative SNR and map these changes topographically across the scalp. So we recruited 23 experienced meditator, both male and female, with a broad age range and diverse background. Each had significant experience with Kriya Yoga, ensuring the reliability of our meditation EEG. So along this EEG acquisition, we did some questionnaire also. That is prerequisites for EEG experimentation. That is GHQ, STI, and BDI. So participants underwent several psychometric assessment, which I have already shown in the previous slide, to evaluate their mental and physical health. EEG was recorded during a 10 minutes meditation session with 4.17 minutes of artifact free data analyzed for connectivity. So EEG was recorded using a 64 channel system with a standard electrode montage. For connectivity analysis, we focused on 8 electrodes along the midline, covering frontal, central, and parietal regions. So pre-processing included bandpass and notch filtering to remove noise and power line interference, normalizations to remove baseline drift, and wavelet-based artifacts removal to ensure clean data for analysis. We modeled EEG data using a time-varying multivariate autoregressive model, which allows us to capture dynamic interactions. The directed transfer functions and PDC, partial directed coherence, are frequency domain estimators derived from these models, both grounded to the Ganger causality theory. So, the DTF, directed transfer function, quantifies the directional flow of information between brain regions, while computationally efficient, and it can sometimes be affected by indirect connections, leading to potential misinterpretations, especially in the presence of volume conductions. And another thing is that the partial directed coherence is basically designed to measure only direct causal influences during the risk of spurious connections. So, however, it is sensitive to amplitude scaling, which can affect the interpretation of connectivity strength. Okay, so this slide shows the visual inspection of the topographical plots that reveals the PDC consistently shows higher intensity and clearer patterns for connectivity estimations, particularly in the frontoparietal regions compared to the directed transfer functions. So here the color intensity or color bar shows the a higher color intensity shows the higher connection, and lower intensity or blue color represents the connection or information flow is less. Okay, this slide shows a typical meditator's directed transfer
I
functions.
couldn't understand the topographical, the topo plots. Can you explain them again?
In the previous slide, Professor.
Yes. What is the red? What is the blue?
Yeah, so red signifies the higher connections. The color intensity is like 0 to 0.12. Okay, 0 means there is no connections or the information flow, neural information flow is very less, whereas 0.12 means higher intensity, means those regions, frontoparietal regions, are highly connected during meditation. So these are just a mathematical measures to show the neural information flow during meditation. So both are based on the frequency-based measures, the partial directed coherence, and along with the directed transfer function. Whereas in case of DTF, you can see that that information which we obtained from the DTF is very less. It doesn't signifies any connections in case of DTF. So most
Oh,
of the regions are
you
blue.
are plotting the degree centrality, or you are plotting what uh, the value of the PDC with what? You have put some seed. This is what I understand, because the PDC, the partial directed coherence, is actually a matrix between the what you say here, the eight selected electrodes. Okay, it is 8 by 8 matrix.
Yeah, it's a 8 cross 8 matrix. That is correct because I have considered only eight electrode.
Electrodes. Okay,
Yes.
yes. So in the area that I see red, for example, in the prefrontal cortex, what this indicates?
This indicates the deep meditations, which is activate the prefrontal region of the brain, and
Yes.
gradually the information is flows towards the frontoparietal regions to the left temporal regions.
Yeah, okay. But you say that you saw in the, as I see in the caption below, you say that there are topographical changes in the partial directed coherence.
Yeah, it shows the gradual transitions, how the information gradually changes in different time instances during meditation.
I don't
So
know
you can see,
how you export these diagrams, but okay, you can continue.
yeah. So to to support your questions, just to ensure, I mean, these are the different time instances topographical representation during meditations. These are all during meditations, and I have shown it's one of the subjects results, because it is we are using 23 subjects, and it's be difficult to uh, so all the 23 subjects plots, but more or less most of the 90, I mean, out of 23 subjects, 90% of the subjects shows the similar pattern during meditations. So initially the frontoparietal regions of the brain is activated, and gradually it will move to the left temporal regions. So this transition shows the information flow or neural activations during meditations. Okay,
Okay,
so
thank
this
you. You can
slides,
continue.
sorry, sir.
I said thank you. You can continue.
Yeah. The slide shows a typical meditator's directed transfer functions and partial directed coherence in a topographical representation. The color bar represents the scale of intensity of information flow. Higher information flows indicate higher connectivity in a specific regions. And so to objectively compare the directed transfer functions and partial directed coherence, we computed absolute energy as a measure of total connectivity, that is signal to noise ratio to assess robustness to noise, and relative SNR, signal to noise ratio, to directly compare the two methods. Which it's just a comparative study, you can say. We are comparing the both the frequency based measures, which one is better to visualize in case of meditation. So absolute energy is also I mean, it's mathematical calculations by summing up the squared magnitude of PDC and DTF values across time and frequency, so providing a measure of overall connectivity strength for each of the estimator. So here we have shown the absolute energy with respect to the number of subjects, like 23 subjects. And in most of the cases, the PDC's energy, the information content in case of PDC, is higher. So this is SNR and relative SNR, signal to noise ratio, quantifies the ratio of signals to noise, indicating the robustness of the connectivity measures. And relative SNR compare the two estimators, treating one as an ideal and the other as noisy, to highlight which provides more reliable connectivity information. Okay. So here in this slide, we generated topographical maps of connectivity at multiple time instances. And DTF tended to highlight parietal occipital regions, while PDC showed stronger frontoparietal activations, indicating higher and more focused information during meditation. So these are like different noise levels, like no noise conditions with a little bit of 5 dB noise, 10 dB noise, then 15 dB, 20 dB, and gradually so and so forth. Similarly, in case of this is the DTF, whereas this is the PDC. PDC gives us more information regarding the neural information flow in comparison to the DTF. Okay, so quantitative analysis across all the subjects shows that PDC consistently yield higher absolute energy, SNR, and relative SNR values, indicating superior performance in capturing directed connectivity during meditation. And this statistical test confirms the difference between PDC and DTF are significantly distinguishable in case of PDC and DTF for to validate the robustness of our findings. Okay, so our findings demonstrate that PDC is a more efficient and reliable estimator for directed connectivity and less affected by volume conduction. And meditation appears to activate a frontoparietal network supporting previous research, which demonstrated yesterday. Using quantitative measures help reduce the subjectivity inherent in visual inspection. Okay, there are some limitations in the study. Our sample had a gender imbalance due to participant availability. We did not include a control group or a non-meditator group and in this study basically. And our findings are specific to Kriya Yoga meditations, which may limit general eligibility. So, okay, future directions. Future work should address gender effect, including non-meditator control groups for the comparative study, and compare different meditation techniques too. Like there are so many meditation technique like transcendental meditation, Chinese meditation, Qigong meditation. So we can include different meditation technique in this study to explore additional connectivity measures and that may further enhance our understanding of meditation's effect on brain networks. So in conclusion, we found that PDC outperforms DTF in assessing directed connectivity during meditation. So meditation induces distinct parietal connectivity patterns and objective and quantitative measures are crucial for accurate analysis. So we thank the Ministry of Human Resource and Development Center for funding this project and all the participants from Hariharan and the Gurukulam Ashram to participate in this experiment and our research team for their valuable support. So these are the couple of publications out of my thesis, the same meditation study. And these are the, thank you for your attention and I'm
Thank you
happy
very
to take
much,
any.
Lakshmi. We have one question from Christos Teriadis, from Mr. Teriadis. Yes, Christo.
Hello, Lakshmi. Thank you very much for the very interesting presentation. I have one question, actually two. Maybe that was mentioned and I didn't catch it, but in what frequencies did you compute it, the functional connectivity, the effective connectivity? Were there specific frequencies or it was in broadband data?
Yeah, actually, you're talking about the data acquisition part or during the uh,
No, during the connectivity analysis. So,
okay,
did
okay.
you choose specific frequencies or was it in the broadband? Because I
It's a
know that
broadband.
meditation may. Okay, okay, yeah.
Yeah, because we didn't focus on particular frequency to analyze the brain connectivity estimation because but we can always see the patterns during meditation. So what frequency is more during that time. And for data acquisition, that is 256 hertz sampling frequency we use to acquire the data. But for the analysis, we didn't focus, but mostly it is 0 to 30 hertz, not beyond that one.
Okay, okay, thank you. And one more question. What MVAR model did you use? Because also the model order, in the analysis, like a usual model order for MVAR analysis is 10, for example. This is the default if you use field
Yes,
trip,
yes.
for example, it's the default is 10, but depending on the model order, you sometimes in some studies you see changes in the results that you actually take. So maybe because you are also comparing between techniques, maybe if you chose a different model order, would that expect to make a difference in the
Uh,
results?
no, for this multivariate autoregressive model, MBAR model, the model order I have restricted to, I mean, that is the optimized model order which we have used, that is 8. And we found that
Okay.
optimized model order P, if you go through my papers, I mean, it has already been published actually. And how we have found out the optimal model order for MBAR. So that is another
Okay.
separate paper for that. I mean, how to find out the
Ah,
optimal
okay, okay,
model order.
perfect. Some kind of criterion then, some kind of criterion for finding the optimal
Uh,
model order. Okay, thank you. Thank you very much. Nice talk.
Thank you so
Thank you.
much, Christos. Thank you.
Manusos, we can't hear you. You are muted.
I'm sorry, I always forget to unmute myself. Lakshmi, thank you very much. It was very interesting. I want to see deeper, so I'm going to read your papers a bit because I was very curious about how you make these plots, these type of plots. In any case, so let's have a break for 30 minutes and then we come back for the last presentation of the Neuroscience Week for 2025 from Dr. Pinotsis. So, thank you very much. See you in 30 minutes.
Yeah,
Okay,
thank
bye.
you. Thank you so much.
Hello again. Let me see who is online. Hi, Dimitris. Why I don't have a sound? Just a second. Probably it's my fault.
No. Hi, Manousis. Good to see you.
Now I can see. I can hear you. Good to see you also. How are you?
All good. Yeah.
Nice,
Everything's
fantastic.
fine.
So let's begin. It's the last session of the last presentation for the Neuroscience Week that we have organized, and we have the great honor to have with us Dr. Dimitris Pinotsis. Dimitri, in which university you are currently? In MIT and in
It's
UCL?
University of London, City St. George's, University of London.
Correct. So, Dimitris, a neuroscientist with very remarkable research, let's say, and today he's going to talk to us about the cytoelectric coupling hypothesis.
Thank you so much, Manousis.
Thank
Can
you
you
very
see
much.
my screen?
No.
I need to share it, I guess. Okay, let me try sharing it. Let's see how it works.
Yes, share it, the entire screen.
Yeah. Can you see my
Okay,
slides now?
yes, can you try to change your slides? Because some,
Yeah.
yes, okay, it's perfect.
Okay, fantastic.
Thank you very much.
So I'd like first to commend Manousis and the team for organizing this. I think this was missing. And also thank Manousis for the kind words. Today, I will talk about some line of work we have been doing over the past several years that somehow culminated into a suggestion for a new principle for brain organization, for which we created some sort of new word deriving from Greek, cytoelectric. And this is an extension of something well known, but maybe a little bit neglected in the neuroscience literature, called ephaptic coupling, another Greek word invented by a Greek, by the way, a neuroscientist in the 1930s who was working, a female neuroscientist who was working in France. Arvanitaki is the name. So what is ephaptic coupling? Ephaptic coupling is the idea that somehow neurons, when they spike in the brain, they are affected by the ambient near field, electric field, by the overall electric field. But this kind of coupling is normally considered to be an epiphenomenon. So something that we don't pay too much attention at. Now, what we said is that the field, first of all, that affects neurons doesn't come only from neuronal activity, but comes from other influences, like something called the cytoskeleton, which is the thing that binds neurons together and contains lots of biochemicals. And this is not just an epiphenomenon, something minor. This is the controlling, let's say, the controlling mechanism for forming memories and even thoughts. So that's what I will talk about today with you. I will talk about this hypothesis and what sort of evidence we might have, and also how we test it in real data, focusing, of course, on neuronal activity and on the representation of information at the level of electric fields. So there is converging evidence, right, about the importance of, in a sense, mesoscale electric fields for cognition. So we know that cognition depends on the mesoscale organization of neural activity, and one sort of evidence for that is the existence of cognitive maps, which are mesoscale patterns that somehow structure knowledge. And we have plenty of examples. The most well-known, I put some examples here, are kind of maps in the hippocampus in the form of play cells or grid cells or other cells that code for various patterns of knowledge that the animal might be having, and people have really observed this kind of behavior. Another thing that we know very well is, and you've been listening, I guess, throughout this week, is functional connectivity and also oscillations, oscillatory behavior. On the left-hand side on this slide, you see data from EEG, and the different colors correspond to the contribution of different frequencies, delta, theta, alpha, etc., to the overall electrical activity at each location of the scalp. So this is the brain, this is EEG, electroencephalography data, and then you have how the power in different branches is distributed across across the scalp. On the right-hand side, you see, of course, the connection between these frequency bands and different sorts of cognitive behavior. So gamma, for example, at the top is linked to attention or mental sharpness, on eye movements, etc. And then beta is, as we might have heard, is linked to prediction, for example, and expectations and wakeful level of consciousness. And alpha is, for example, is more occipital, associated with rest. Theta with planning ahead or even drowsiness and unconsciousness, or is used for neurofeedback often and meditation, etc. So there is cognition linked to these mesoscale patterns of activity. Again, and when we also record, let's say, neurons in individual trials during a perceptual decision-making task or a navigation task in a maze, we know in different days that the neurons that represent information in such a task change, something known as representational drift. So if you look now on the right-hand side here, on this panel, you see a bunch of neurons, and some of them are purple for day 1, and on day 2 at the top of the right-hand side, you have a different bunch of neurons. But then when you look at the manifold on which neural activity is projected, and this is supposedly what carries information about the task, and using this readout from that manifold, we can decode information for processing at later stages across the cortical hierarchy. We see that the manifold is stable. So neurons have been, let's say, are thought to be similar to ions in a crystalline structure in a metal. So in different trials, they are different. They come and go from the neural ensemble that represents information, but somehow the overall structure is maintained. And this is basically the suggestion here is that the electric field somehow, or the energy state, is stable, but neurons can move freely in and out of the neural ensemble of the memory pattern, as we'll see in a minute, similarly to ions in a conductor or a metal. So what are these neural ensembles? What are these neurons doing? We have experiments for example, optogenetics and also from optical imaging, where, let me mute this, where we can map them, right? So what you're going to look at now is a video from a mouse that is on a treadmill, it's running, and also is navigating itself and looking at some images within a virtual reality environment. There is a screen in front of it, and then this microscope on top of the head, it's measuring the neural activity spiking. So you will see different neurons spiking in succession, like synfire chains. And this constitutes a neural ensemble that maintains information about the visual stimulus that the mouse sees. And if you show the same sort of stimulus, you'll have the same neurons being formed, more or less, again, with some drift. They will come and go, but in different trials, it will be more or less the same part of the cortex. And you will see, where is this? You see here, essentially, that there is activity of neurons, and you see at the top here the cells in different, sorry, at the bottom, cell number 1, cell number 2, cell number 3, different colors, red, blue, and green, and they spike in a certain succession. So more or less the neurons are the same, but not exactly the same, of course, and together they form something called neural ensemble. On the right-hand side, you see here data from an electrophysiology study. This is a study where the monkey now had to apply a certain rule, and this is the monkey brain near the posterior sulcus, which is given by this line, and you have these dots, and these are electrodes. So this is a multi-electrode array that covers a large part of the monkey brain, and for different rules that the monkey has to implement for the task, you see different pairwise correlations. So each line on this plot is a pairwise correlation between two electrodes on the monkey brain, and depending on the rule the monkey has to apply at its essentially at its trial, the pattern of these pairwise correlations stays the same. So it's not only about let's say sensory information that we have a formation of ensembles. The same neurons will fire together when the monkey uses even an abstract concept like a rule. So for each trial, we'll have more or less the same pattern. But of course, neurons again will be slightly different, but the overall correlations will remain sort of stable. So what is the converging evidence, right, from all these studies, for the role of mesoscale patterns of electrical activity, is that there is some sort of stability emerging out of the spiking of a bunch of neurons. So neurons generate an aggregate mesoscale electric field. You can call it the electric field of thought neurons, let's say, about thoughts. And then this electric field somehow constrains these neurons and makes them form ensembles, these neural assemblies that you just saw on the previous slide. So that this feedback kind of mechanism. And this is precisely the idea for cytoelectric coupling, that somehow this aggregate electric fields that originally is produced by individual neurons then feeds back to neurons, but also other structures in the cytoskeleton. And there's a goal here, right? The goal is to tune the cytoskeleton, to tune the neurons and the proteins, the microtubules, the filaments, all the biology that binds neurons together, the extracellular matrix, to process information more efficiently. So you can think of an analog like a conductor of the orchestra. This is the electric field that somehow guides individual musicians and different sections of the orchestra, and also provides stability, right? Is what really creates the orchestra itself. Imagine having an orchestra without any conductor and maybe 60 or more musicians. It will be chaos. It's impossible to function. So same thing about the brain. So let's see what sort of implications, first of all, this might have. So what we know from, let's say, the classical neural code principle is that spiking carries information, and spiking is all we need to explain information processing. So for example, in sensory areas, we have a very simple case here. On the bottom, on the top left, this A panel shows what? Grading patterns, different colors of the boxes mean different orientations, 0, 30, 60, etc. degrees. And then the corresponding activity in primary visual cortex, for example, is shown on the bottom panel here, C. You see the red spikes and the yellow, etc. So there is some kind of mapping between what you see and basically what is coded in the spiking activity. Manouchios, I see you up. Is there something? Is there a question or something, or I'm missing? Or can I go on? No. Okay. Very good to have you here.
Please continue. Yes. No, I was here, but I thought that I opened my camera, but it was closed.
No problem. Just let me know if there's something, because I'm not sure I see everything in the Zoom. Yeah. So what is the idea, right? What is the neural code principle? Is that if you take now these spikes, so the responses of neurons as units, you can decode all sensory information, all these grading patterns, for example. So spiking is sufficient. That's the old idea. But if now with the new idea, in a sense, the field has a more stable and guiding role, it means that the level of information representation at the mesoscale, at the level of field, is distinct and maybe even more powerful than spikes. So this is the cytoelectric coupling hypothesis, that spikes are not enough, that the emerging electric field contains stable information representation and guides neurons and the other attendant structures to process information more efficiently. So what is evidence so far? And we have some unpublished work, but I'll talk more about published work here now for this hypothesis. And by the way, on the right-hand side here, you see an example of what the microskeleton really is basically you have the neurons, you have the neuronal membrane down here. Maybe you can see my my pointer. And then you have these other structures, right? Within neurons, you have these microtubules, and if you like, consciousness studies, for example, and you are an aficionado of Penrose, and you read the book, Shadows of the Mind, Skiestounou in Greek, you might have heard about these structures that are associated with the emergence of consciousness. So these are the microtubules here within neurons, but then you have, of course, what binds neurons together, like I was saying earlier, the extracellular matrix with proteins, tubulin, and others. You have intermediate filaments. All these are charged structures. So these are generating, but also affected by the ambient field. And this is the cytoelectric field that we are talking about. So let's take a step back and tell you exactly like what the setup is, what we know in state-of-the-art experiments, and then our work, how to, let's say, find evidence for this suggestion about the importance of electric fields. So a classic way to look at memories, right, is in small animals, like we saw earlier, like rats and mice, and you put them in two cages, the white cage on the left, the context A cage, and the context B, and then you associate sort of memories, you build memories in the rat or mouse brain by somehow punishing, for example, the animal in context A, giving it electric shock, and giving it food in context B. And so you form two memories here. The left memory, the white memory, let's say, context A is fear memory. The context B, the blue cage memory, is the pleasant memory, the reward. So what can you do once you have these memories and you've induced them in the animal brain? You can now go and look which neurons, similar to the optical imaging example we had earlier, for example, or optogenetics we saw in the previous video. You can look which neurons form these memories, where in the brain these memories lie. And then there's a pattern, a mesoscale pattern, again, of effective connectivity associated with this memory. And what is remarkable, and people have done these experiments like in the Kotonegawa lab, for example, at MIT, is that now you put the animal, after they have formed the memory, in the safe, in the blue, the reward cage, and you activate with optogenetics the fear memory, the memory that is associated with the context A, the white cage. And then what you see is that the animal responds, although it's safe, is in the blue cage, it recalls and it uses the fear memory. So you can control essentially which neurons, so you know that these neurons, when they're activated, they somehow bring up the unpleasant feelings that you had in the other context. So the bottom line here is that there is a mesoscale pattern of effective connectivity associated with this memory. So this is what you can also do in larger animals like monkeys. Using our computational modeling, we can find this microscopic connectivity of neural ensembles. So in the data I will show you is an old data set from MIT, from Miller's lab, where the monkey was trained to decode saccades that's used for oculomotor brain-computer interfaces. It's a useful thing to train the monkey on. So what is the paradigm? Very simple. The monkey will respond with a saccade, which means it first fixates. There is a cue, as you see here on the panel, at a certain location in the visual field, at a certain angle in this case. Then there is a delay, and the monkey has to respond by remembering and saccading to the location that was presented to them. So they somehow, when you have a paraplegic, for example, one way to communicate information is by doing these saccades. And the monkey, if it remembers the correct location here, the green dot is happy, and when it's and gets a reward, and when it's incorrect, it does not. So how can we find now these microscopic patterns of effective connectivity? We use something called the neural field model, which basically predicts activity on a cortical patch. So it describes spatially extended neural processes in the brain, and basically you can get predictions of the sort of activity you expect to record if you have a multi-electrode array, and different colors here are different levels of depolarization, and this is a small patch, let's say 3 by 3 millimeters on the monkey cortex. So we have this model, and we can apply this model to predict the different neurons that participate in each neural ensemble, in its memory, and we can get now the connectivity of the neural ensemble, this microscopic connectivity, by training the algorithm using LFP data. And this matrix is what essentially tells us is the second bullet point here, how much signal is propagating from one electrode or suboculation of the ensemble to the other. So the axis here, 0 to 30, etc., is the number of the electrode on the multi-electrode array that sits on top of the monkey brain. Sorry, similar, for example, to what we had. Sorry for that. Let's see. Yeah, that's the picture we have, right? We have this multi-electrode array. This has numbers, and then we get basically effective connectivity, which is similar to functional connectivity in these matrices up here. So we have location by location, and the entries here are exactly the connectivity weights. And what we see is that in a data-driven fashion, in frontal areas in this case, so we record from FEF that somehow has a visual representation, a visual map of the visual field, because it's used for the saccade command, to generate the saccade command. We get this translational invariance that we know exists in sensory areas and also in, for example, convolutional neural networks. So the interesting thing here is, number one, that we got this kind of convolutional structure in a data-driven fashion. We didn't impose it as people do in convolutional neural networks. And second, this comes from a frontal area, not a sensory area, that again, like I said, is known to basically create a map of the world to use for saccade initiation. And you can validate this. I don't have time to talk about. So you can validate really the microscopic connectivity using SVD, for example, some sort of high-dimensional SVD. You can, or clustering approaches like K-means clustering, and that's what we did. But now, why is this useful, right? Remember, I want to talk about electric fields. So now I have, let's say, the ensemble or an in silico, a digital copy of the neural paths, and then I can put on top an electric field model. So I will have two models now. I have the neural field model that describes spiking activity, the first level of information representation. And then I have now the electric field model sitting on top that describes the projection of spiking to mesoscale patterns of neural activity. It's like oscillatory patterns at the mesoscale, similar to LFP, but a little bit one step, let's say, above the LFP. And basically what we did is we used a very simple model for the electric field, similar to Newton's law in the gravitational field that you might remember from the school, from your secondary school. So you have the masses, right? M1 and M2, like the Earth and the Moon. You can get the force, and if you have the force, you can get the gradient, the field out of this. Similarly, for neural activity, you have the neural field model for spiking, and you get now the electric field using the analog for electromagnetism, basically, of this. And that's the model we use. It's called bidomain model, and we make some assumptions, of course, of homogeneity. So you take the pyramidal cells that are somehow aligned throughout layers 2 to 5 in the cortex, and you create this kind of basically the cylinders, for example, like looking at activity that propagates along these long actions, and you take now the current, and once you have the current and the dipole moment that generates electrical activity, you can get extracellular potential. So what is basically the near field? If you have the pads and you measure with the electrode on top of the pads, what is the field that exists there? This is an extension of an idea used in cardiology. So this model was used first for getting the electric field that you have in a cardiogram, for example. And what is interesting, right? Since you've been, I guess, listening to talks about function connectivity at the mesoscale or large brain organization and default mode networks, etc., that this micro-effective connectivity is a biomarker in the sense that you can decode memories from FEF, for example, on the left here, based on the connectivity patterns. And what you see on this plot is basically the output of different classification algorithms, diagonal LDA or naive Bayes. And the vertical axis here is accuracy, essentially. And the different colors correspond to different basic connectivity patterns. And because of how the algorithm works, we have different sorts of, in a sense, connectivity components that are similar to PCA components, if you will. If you have any more technical questions, we can discuss it later. The point here is that if I know the microscopic connectivity pattern, I know the memory. So this is some sort of decoding feature that I can use to decode with above chance. As you see here, it goes up to much higher, double accuracy compared to chance level, which is shown here with the dashed line for this naive Bayes classifier. And this sort of greenish, this sort of greenish bar, number 2, with naive Bayes, and this is the dashed line I was talking about before. Manouchios, how much time do I have? So I don't want to overrun too much.
It's about 7 minutes more, but you are free to, because it's very interesting, your presentation, you are free to go as you like.
Okay, then I won't rush it too much, sorry, because it's good for people to be able to follow. Yeah, thank you for giving this. Okay, so now the other interesting thing is that not only the micro connectivity, in a sense, the micro-effective connectivity is a biomarker in the sense I described, but also the electric field that corresponds to each of these basically each of these neural ensembles is also a biomarker. And again, what I'm showing here is the result of decoding and the decoding accuracy on the vertical axis. Again, the horizontal axis is, in this case, the electrode location. So the value of the electric field at a certain location of the patch or the electrode that we're measuring from. And again, this is above chance. In the case here that the orange bars, sorry, the blue bars are larger than orange. This means here that the observed accuracy is significantly higher than chance. So you see for a lot of locations, this is the case when blue is larger over here. And what is the interesting thing? The interesting thing is that if I look now in the decoding accuracy of the field as opposed to neural activity at each electrode, this is higher. Essentially, this you can show using a Velk t-test. So that somehow there is more information, if you will, at the field compared to spiking. That's an interesting result that, in a sense, is sort of converging evidence to this cytoelectric applica hypothesis. That's number one. Number two, remember I said the field is like a conductor. What does a conductor do in an orchestra? It provides top-down guidance and stability. So let's look at guidance first. The way we look at guidance is by now taking our field estimates, right? We know what the field is at its location. We know what the spiking is through our neural field model. And we look at gradient causality between the two. For those of you who don't remember, gradient causality is an information theoretic measure that basically says, if I want to predict the value of a variable A, given past knowledge of this variable, right, the values at preceding time points, is this somehow, this prediction, does it improve when I consider not only the past values of the variable that I'm looking to predict, but also the past values of some other variable? And if this other variable also helps predict the upcoming value of variable A, let's say variable B helps predict the future of variable A, this means that there is an information propagation transfer from B to A. So B affects A in the future. So somehow information from B is transmitted to the future of variable A. So that's what we did here. We said you have the electric field predictions or the time series for electric fields. You have the spiking predictions, right? You know what the time series is. If I want to predict the upcoming information for spiking, does it help to bring in information of the field? And the other way around, does the prediction for electric fields improve when I put in spiking? So it's both ways. So what you see here, let me focus maybe on the panel C, E, etc., the red and blue. So the red and blue bars here, let's focus on that in the interest of time. On the horizontal axis, we have milliseconds, right? In this panel, let's say C, the red bars. And the vertical axis is Granger causality, what I just described with a twist, that we didn't use the classical Granger causality, but if we have time, can talk offline or later about it. So field to activity means that what is the impact from the electric field, the mesoscale, to the micro spiking. And the impact, let's say, is quantified in terms of these values, this Granger causality coefficient, which is like an F-test, log likelihood ratio between the models I just described with variable B or without. And you see, let's say, this is of the order of whatever, 8 or 7 here. And then you do the same game, you play the same game in the reverse direction. These are the blue bars, and this is for FEF, for recordings in FEF at a certain location, right? At a certain ensemble, 120 degrees. This is the theta you see at the top here. What can you see immediately? That is an order of magnitude difference between the blue bars here and the red bars. So the red bars are about 7 or 6 to 8, and the blue bars are 0.02 to 0.06 or 0.8. So it's huge difference, right? This is immediate. And this, again, same area, same ensemble, same memory for theta angle equals 120 degrees, but the reverse direction. And we also have other angles here in other plots and in another area as well. So what do we see across all these plots? That the field to activity is larger than activity to field. Like when I say activity, I mean spiking. So field to spiking is larger than neural activities and spiking to the electric field. And then if you look at because you see all these blue bars are smaller. And if you look now at the temporal variability in these Granger causality measures, and you quantify this using this coefficient of variation, what do you see? You see that the variation for the red bars is very, very small, 2 to 4%, as I wrote here. This is panel K. Coefficient of variation is on the vertical axis, different acute locations and angles are on the horizontal axis down here. So the red bars in panel K at the bottom right, they're much smaller than the blue bars, which are about 28 to 47%, the coefficient of variation. So this tells me that the field is more stable also, while the other panels told me that there is top-down guidance, right, from the field to the spiking. Some other results also, again, arguing in favor of this top-down guidance. It's again, here this is similar, Granger causality. Again, I'll focus here on panel C, since we don't have time. Time is on the vertical axis and Granger causality coefficient is on the time on the horizontal axis time, on the vertical axis Granger causality. What you see here is that essentially you have now two areas, right? You have FEF to SEF field in panel C, the red panel here, and SEF to FEF field. So the top panels here for the same memory ensemble for 120 degrees, you have this panel C, and then you have neural activity at the bottom. And immediately you can see that the Granger causality at the level of neural activity is sparser, right? You have 0 values or very, very small values at the this panel here for neural activity for both directions, FEF to SEF and SEF to FEF in blue here, compared to the top panels. So it's sparser. So what does it tell me? It tells me again that there are more influences, more stable influences from the field at the level of fields between two areas now, compared to neural activity that is 0 influence sometimes. So nothing is happening. There's no communication in a sense between the two areas. And this is, by the way, is similar to communication through coherence, this hypothesis that Pascal Fries and others have put forward. So this is another, let's say, argument in favor of top-down guidance for the fields. And again, for stability, right? How can you measure stability? Very simply with correlations. That's what we did. We found now correlations between different trials for the same memory and for the same, of course, location on the cortical paths. And you see immediately here that in yellow, you have correlated trials in these matrices. The dimension of the matrices trials by trials. So if you see a yellow entry here, it means that these two trials are very, very similar. And the number of yellow entries on these matrices for fields on the left and neural activity on the right is consistently higher when you look at the electric field matrix. So electric fields are more similar across trials compared to neural activity. Of course, this is a memory paradigm, right? So there is some correlation across trials. There is some overall stability, but it's more stable on at the electric field level. That's another piece of evidence that we have. And the last piece of evidence, follow up from the previous slide, is that the percentage of electrodes with significantly more correlated EF estimates is larger, and this is replicated here using a Fisher exact test across different locations and different angles. So that's basically at least a recap of some evidence that we have so far for this hypothesis, and I showed you experimental, let's say, data and some computational analysis that argue in favor of both guidance and stability. So yeah, the take-home message is that somehow you have emergence of electric fields, and this is similar. I'm not sure I could. Can you see now? You cannot see my. Let me show one last video just for you to remember it. And for this, I need to somehow stop a little bit my share because the video doesn't work, and use YouTube. 1 second. Let me reshare my screen with that YouTube video that I had put on the slide, but doesn't work. Yeah, you can see now the YouTube video, right? Yeah. I hope.
No,
Okay,
we cannot see it.
You cannot see it. What can you see? Okay, 1 second. Something is not working. Okay, I'm taking a little bit. What's going on? I don't see the controls here. That's very weird.
It's below in the screen. Click with the button in the screen, your face.
Yeah. Okay. Yeah.
Probably
Yeah. Somehow. So let's see. What can you see here? Now you see a black screen. I don't know what
black
happened.
screen, yes.
Yeah. Okay. So anyway, I'll wrap up here so we don't take too much time. Something happened in the screen and somehow it's not. 1 second. Anyway, since I promised that, let me give it another try. You can see it
Okay,
now,
now
right?
we can see that, yes.
Oh, okay, let me put it in. So what is the analog here? And this is an experiment you can do in your kitchen, by the way. So what you can do in your kitchen is take a frying pan and pour olive oil in it and basically start heating it, right? Heating the pan. And what you will observe, if you had a microscope, is what these guys observe. So you will observe some hexagonal, for example, structures, if you like chemistry at school. So what happens here is you have temperature, right, changing in the frying pan because you have heat underneath. And this changes how molecules of the olive oil come together. So they form these ensembles, if you will, through self-organization. This is well known in complex systems theory. And depending on the heat,
Dissipation systems.
this is called convection instability.
Mhm.
So this is the idea here. The idea is that somehow neurons fire together when you have input coming from sensory areas, for example, when you have to recall. Let me stop this. And somehow they organize together, and this feeds back and helps this process in a feedback loop. And this is very crucial and have implications, for example, for building efficient brain-computer interfaces, which is something that we are trying to use now, all this theory for you to do. And with this, let me reshare my screen just to show the thank you slide, my colleagues at MIT and at Johns Hopkins. Okay. So, and also, yeah. So this is a thank you slide. So thank you all for your attention. And we're going to have
Thank you
questions.
also, Dimitri. Are there any questions? Until someone or something, I would like to ask something. If you go to the slide that you show the 3D, what it was, landscape, let me tell it, the electrical field plot, the 3D electrical field, where you had...
Yes. Yeah, you're right.
Okay.
Let me bring it up.
To be honest, the last year I'm working or something similar in energy landscape, or in energy landscape, exactly. So I was the previous slide, the previous of that. So you have the micro array of connectivity of the micro array, let's say that, and then you have the electrical field, and you see these models actually and the interactions. Have you considered or have you tried, I don't know, to incorporate here also thermodynamics in the sense of energy landscape or the gradients of the free energy principle, free energy of whatever is here?
Yeah, so the way supposedly this works is, I mean, people have used, let's say, Cohen maps and self-organization to describe the formation of retinotopic maps, for example, or numerosity maps. So yeah, if you assume that you have its input driving, let's say, cortical paths, right, and this happens also during development, then the emerging activity will feed back, and of course the impact of the input will feed back to the whole ensemble and create these sort of structural patterns. There are also biological processes, by the way, talking about the cytoskeleton that help that. There's something called mechanotransduction. And also there are these mechanosensitive proteins. That's why the cytoskeleton is important. So you have input coming to a sensory area, for example, during development. And then the mechanical energy of the input is transformed into electrical energy, and then it feeds back and tries to organize neurons. So yeah, I mean, the direction you're suggesting is learning is development, and this is a very interesting, in a sense, open kind of problem.
Okay, that's good. It's good to hear. Okay, any other question, please? Not so far. So, Dimitri, thank you very much for
Thank you,
joining us. It
Manos.
was an honor and pleasure to have you with us. Let me close this event. Thank you very much, everyone who took part in this, let's say, week full of neuroscience. I hope that everyone learned something new, and most importantly, you feel, let's say, the the spike of curiosity and let's say of eager to learn more about neuroscience and study more about neuroscience. So anything you need, you can come in touch with every one of the presenters or on me, and I can redirect you. Thank you very much, everyone. Have a nice day.